All right, hey guys, so my name is Max. I'm one of the SIs for Dr. Maduro's 5A class. And today we're going to be going over these two questions from past midterms that Dr. Maduro has. And they're both about cell respiration. And they kind of follow the same thought process about going about them. This is the way that I would approach it. And I just wanted to share that with you guys. All right, so the first question asks that we have a diagram. It's an animal cell. All right, so that's something to note. We have an animal cell, and we're referring to this diagram, and it's numbering different parts. And I'd say the most important thing here is to first figure out what's everything really referring to, right? So let's look at location one. Location one, which is right there, this you can infer to be the nucleus. That's because it is part of the endomembrane system, if you guys remember that, where it's connected to the ER, or the endoplasmic reticulum, as you guys see right here. And so we can think about what happens in the area. Generally, you have DNA replication, you have all that type of stuff, anything to do with DNA. Nothing much really in terms of our cell respiration processes like glycolysis and the Krebs cycle. So we're gonna move on from there. Location two, it's the kind of the empty space everywhere. And so you guys would know that to be the cytosol or cytoplasm, same thing. Right, and so what happens in the cytoplasm that you guys know? For sure, we know that glycolysis happens there. And we know that pyruvate oxidation also. Right, next, let's look at location three. And so that's referring to this space right here, and I will try to fill it in. So if you guys can see my red marker, I believe location three is this space right there. And the main thing here is understanding that right now we're looking at the mitochondria, and we're looking at the space between the two membranes, and that is the intermembrane space. And what happens here, nothing really happens here in terms of it being in a vacuum, right? It's more involved in different processes, such as bringing in or transport of acetyl-CoA, or we have the electron transport chain that happens somewhere here. So those two things, for example, are in the intermembrane space. All right, number four, we're also referring to the mitochondria, and this would be the matrix, or the mitochondrial matrix. This is just the space inside the mitochondria, and you guys know that what happens in here, this is the Krebs cycle, or you can call the citric acid cycle, or we can say it's the tricarboxylic acid cycle that I will abbreviate as TCA. All right, and lastly, we have five. And so five, again, it's part of the endomembrane system. It's after the endoplasmic reticulum, or the ER. And so this refers to the Golgi apparatus, Golgi body, any of those is fine. And so when we think about what happens here, this is more for packaging materials that will leave the cell, right? So in terms of which ones we're not concerned with so much in this problem are the ones that we don't really deal with for cell respiration, right? So that would be, for example, location one, we don't really deal with, and location five, right? So these are both somewhat out of our answer choice here. And so we would then look for the rest. So we have 2, 3, and 4. So we kind of look at the different processes that we have. So in number 3, that says it's lactic acid fermentation. And so when we're looking at this, we know that lactic acid fermentation, this is when we are anaerobic, so we don't have oxygen, and this would happen in the cytosol. So this would actually be in location 2, right? So we know that this doesn't work. We can look at location 1 as being glycolysis, and again, we know that glycolysis, this happens in the cytosol, which is also location 2. So that wouldn't work. Number 2 for transcription, right? Transcription we know happens in the nucleus. That's when we copy DNA to make mRNA. And so this would happen in location 1. And so that wouldn't work. We have number 4 as being the citric acid cycle and 5 being translation, right? So translation, remember that translation is when we go RNA to protein. So this does happen in the endomembrane system, but it would happen on the endoplasmic reticulum. And it's not numbered here. I'm going to write ER, but it is not numbered. And so that would leave us with number 4 as the citric acid cycle. And that is where it indeed happens. We have it here where we said the matrix or the mitochondrial matrix. That is where the citric acid cycle happens. So that would be our answer. All right, next question. So let's look at this one now. Again, similar idea, but this one now we're zooming in on the mitochondria and really looking at the events that take place there. Okay, so the question is, there's a diagram and they're corresponding to solute movement. Okay, so that's a big thing to remember here, where solutes are moving. And from the diagram, where is oxygen most directly required? Okay, and so let's think about that. Again, we want to know what these locations are referring to. So location 1, this would be a membrane complex or a protein that is on the inner mitochondrial membrane, right? What process happens here? This would be at the end of the electron transport chain where we use ATP synthase to make ATP, right? Remember that that's a coupled reaction with hydrogen pumps, or the hydrogen ions moving back in. And then we go to location 2. So what's location 2? This is, again, a membrane complex on the inner mitochondrial membrane, but now it's pushing the solutes out, right? So what's pushing it out? This is when we move hydrogen into the intermembrane space, right? All right, then we have location 3. Location 3 deals with the Krebs cycle, and so this would be the mitochondrial matrix, right? And what process? We have the Krebs cycle. I'm just going to put Krebs, but as we know, there's different names for that. And lastly, we have 4. So what is 4? This is just the mitochondrial membrane, right? So this includes the inner membrane, the outer membrane, and the space. Okay, and so what happens here? This is when we transport acetyl-CoA into the mitochondria. Okay, so when we're looking at this problem, we're thinking about now, where is oxygen most directly required? We know that it's going to be anything that has to do with the electron transport chain, right? And what do we know about oxygen in the electron transport chain? We know that in the electron transport chain, oxygen is the last electron acceptor, or the primary electron receptor. You can call it the receiver. I'm just going to write rec. And so the point here is that we have to figure out, where is it? So we know the electron transport chain doesn't really deal with location 4, right? Again, we said this is for the transferring acetyl-CoA. It doesn't also deal with the mitochondrial matrix so much. So now we're zoning in on locations 1 and 2. So location 1, this is where we said we pump hydrogen ions, right, that were in here. They're in the intermembrane space, right? We're pumping them back into the mitochondrial matrix, okay? And so let's think about that. In this case, we would have a byproduct here, right, where we make, using this arrow, we would couple that reaction to make ATP, okay? And so is oxygen really involved there? You might think maybe it is. However, in this case, it is not. Remember that ATP is made with ADP plus another phosphate group that we will call PI, okay? In this case, with number 2, if we're looking at that one, we know that what's getting pumped out, like we said in our table here, we said that we're moving hydrogen. So we have hydrogen here, and we're moving it outside. We're moving it into this intermembrane space, and we're creating that hydrogen gradient, right? Where the intermembrane space has the high concentration of hydrogen. And hopefully this is clear. This is a high concentration. And in here we have a low concentration, right? And so the point here is that when we move the solutes, we're thinking about what's actually moving. And in this case, our solute would be hydrogen. Where oxygen is required is that once we're moving these hydrogen ions out, remember that oxygen takes in the electron and it ends up making water, right? So we have our 1/2 O2, and we take in the electron and we end up making water. Okay, so again, we wouldn't really be dealing with location 1, or sorry, we wouldn't be dealing with location 2, sorry, we wouldn't be dealing with location 3 just because we're not dealing with anything in the matrix. We also wouldn't be dealing with location 4 just because, again, it needs to just be the inner membrane and the matrix. Next, we also wouldn't be, therefore, be dealing with locations 1 and 4, right? They are, in fact, different. Location 1 or location 2, right? That's where we narrowed it down to. And again, we wouldn't be dealing with location 1 just because this would be the end of the electron transport chain, and this does not require oxygen. Remember that, again, we make ATP at that last step, and so that leaves us with our answer choice E, which is location 2. If you guys have any questions about this, please feel free to reach out to Dr. Maduro or to us as SIs, and we have questions like this on our worksheets that we update for you guys every Monday and Wednesday. All right, thank you.